Yeah, so this abstract was actually on a specific biomarker, which is called TARC, spelled as TARC. And this TARC is really an extremely attractive biomarker in classic Hodgkin's lymphoma. And there are three main reasons for that. So first, it is literally attractive because it is a chemokine, a protein that attracts immune cells. TARC stands for thymus and activation-related chemokine, and surprisingly, it is produced and excreted in very high amounts by the Hodgkin Reed-Sternberg cells, which are the tumor cells in Hodgkin's lymphoma. Secondly, TARC has a very important function in the development of Hodgkin's lymphoma in most of the cases. The Hodgkin Reed-Sternberg cells are present in only a small number in the tumor mass, and single tumor cells are surrounded by a high number of other, more or less normal immune cells. TARC attracts CD4 positive T cells that have CCR4 as a molecule on their surface. And this actually leads to a very characteristic phenomenon to Hodgkin's lymphoma. That is the formation of a cloud of CD4 positive T cells around each single tumor cell. And these clouds are better known as rosettes. The Hodgkin and Reed-Sternberg cells modulate the rosetting T cells in a specific way. And actually, they are changed to provide growth factors and survival signals to the tumor cells. And these rosetting T cells also appear to have another important function, that is to shield off the tumor cells from hostile immune cells that are located outside of the rosettes. And then third and finally, TARC can be very easily measured in blood samples, preferably serum. It is produced only in low levels by normal cells, for example, activated dendritic cells, leading to a low background level in healthy individuals. It is really a tumor cell biomarker. So serum TARC really reflects presence of Hodgkin and Reed-Sternberg cells in individuals. So in serum samples taken at the time of diagnosis in classic Hodgkin's lymphoma patients, we already know that TARC is tremendously increased in more than 90% of cases. So there's a median increase of approximately 400 times compared to background levels. There is no other cancer or other condition known to result in such high levels. So yeah, we took this knowledge in our recent study where we wondered what the dynamics of TARC levels would be in serum prior to diagnosis. We got a hold on serum samples from some Hodgkin lymphoma, some 100 Hodgkin lymphoma patients who had these samples taken in the years before diagnosis. So very intriguingly, we saw that the vast majority of patients had highly increased TARC levels 2 years prior to diagnosis. And going back further, there were patients with increased levels 3, 4, or 5 years before diagnosis. And there was even one patient with significantly increased TARC levels 7 years before diagnosis. This increase in TARC was exponential, and this suggests an exponential growth of the tumor. So this is a very striking finding, and it is important for two reasons. So first, it tells us something about the biology of Hodgkin's lymphoma, which apparently is usually a relatively slowly growing tumor. And secondly, our finding indicates that there may be a potential to diagnose Hodgkin's lymphoma patients earlier, at an earlier stage and with less tumor loads, which would enable less intensive therapy and a better prognosis for our patients.